pleasure to welcome you all to this first webinar series on the theme Coronavirus Pandemic and its impact on human life. I, Dr. Rashmita, Assistant Professor from the Department of Life Science, will be hosting this session today. Before starting the event, I would like to extend my warm welcome to our chief patrons, Honorable Vice Chancellor, Central University of Karnataka, Professor H.M. Aishwarya Sir, Honorable Pro Vice Chancellor, Professor G.R. Naik Sir, and Honorable Registrar, Professor Mustaq Ahmed Patel Sir. Thank you for joining us today. Leaving in its wake near 14 millions of infection and over 60,000 deaths and a huge economic toll, SARS-CoV-2 has devastated the entire world. The continuing spread of this deadly virus remains a public health concern. Scientists across the world are racing against time to get a deeper understanding on the novel coronavirus and develop treatment to combat the infection. Today, with us have joined a group of eminent scientists who will discuss on the recent developments and challenges associated with the infection. I sincerely hope that you will enjoy this session today. Now, may I request our chief guest for opening the event. We are delighted to have our chief guest, Honorable Vice Chancellor Sir, Professor H.M. Aishwarya Sir. He is the man of distinct vision and idol of knowledge and experience. The entire campus is vibrating and echoing his prominence. I request our chief guest, Professor Maheshwarya Sir, to inaugurate the event and address the audience.
Hello, Vice Chancellor, Professor G.R. Naik, the Registrar, Professor Mustaq Ahmed Patel, and the faculty members of the Life Science Department. So I'm very happy that we have the Central University of Karnataka, we have the Physical Science, Chemical Science, Mathematical Science, and other departments. So my wish was to have the Life Science because we doesn't have the Life Science. Many universities in India have the Life Science Department, but unfortunately, this university, even after establishment of 10 years, we couldn't have the Life Science Department. So when I had requested the University Grants Commission and MHRD to start many departments, my priority was the Life Science. So I told them, so that's whatever department you can give, fine, no problem. First, there should be a Life Science Department because the life science is very important for the university because the other sciences are there. And also I was a student of botany, zoology, physics, chemistry in my graduation. So I know that what is the life science and what is the importance of the life science in the university. That's why I requested the committee headed by the Chavan, the former University Grants Commission chairman. He is also being a life science man. He immediately readily accepted. I'm happy this the Central University of Karnataka is having the Life Science Department. So other ambition was that we should have a very good faculty for life science. That is also my intention to select the best faculty members to their department. So my Pro Vice Chancellor helped me. So we invited the best faculty, the best the experts to select the faculty, and we have the. I'm very happy that 4 lecturer posts have been filled. And we could not fill the reader and professor post, and it is yet in the process. So they also may come and join shortly. So that's why we started this life science department. Immediately, that is after this one. So recently, the entire country is facing a lot of problems of this pandemic, the disease, the COVID-19, and all problems were there in Gulbarga, where we are staying here. The Central University is located in Gulbarga. It's only the one university we can have in entire state. So here, so my intention was to have here there is a KIMS, the GIMS was there, and other some 1 or 2 institutions where they are actually the laboratory is there, they are checking about the pandemic diseases. But our department, we thought that when the MHRD and UGC asked us whether we can able to identify the COVID cases like that, then we said yes. because of the four faculty members, the teaching fraternity, we said that yes. So immediately the ACMR and other University Grants Commission, they supported us to start this one. So that is the at present the no grant has been released. Okay, they are going to release and we are approaching the concerned body to release the amount. But we already spent nearly 40 lakhs rupees to establish the life science department. Not departments and to establish the laboratory. We got very important equipments and they are yet to get the safety cabin and other the equipments. We also start the COVID testing, the laboratory in this university. I'm very happy as a head of the institution. They are very happy. So while allotting the building, when Rasmita and other faculty, Rakesh and other faculty members were there with my vice chancellor, he said, so when I can see the virology department in China, so we can also can the name like virology or immunology, something like that. That's it. The science department is there, but for that building we can. I requested them to put this building, and we are not decided. We are going to do that. So here I would like to tell only the brief words, and already our faculty member Rasmita told briefly about the situation of this COVID-19 in the India, what is happening in the whole world. As you know that on December 31st, 2019, a strain new pneumonia of unknown cause was reported in China's, where the WHO country office. The cluster of these cases originally appeared in Wuhan, a city in the Hubei province of China. These infections were found to be caused by the new coronavirus, which was given the name 2019 novel coronavirus, that is called COVID-19. It was later renamed as severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. Coronavirus is highly transmittable and also pathogenic viral infection which spread rapidly around the world. In India, the virus has spread widely and the number of cases have surged to over 10 lakhs active cases and over 27,000 deaths on July 18, 2020. Indian government has put enormous effort to slow down its spread by implementing a proactive nationwide lockdown with the goal of flattening the curve adequately. Also, the government has adopted an intelligent testing strategy called triple uh, triple T. That's a strategy called test, track, and infected and trace their contacts. Strategy to reduce the spread. The number of asymptomatic cases are on the rise. Government has put efforts to create on infrastructure to provide testing facilities and services across the country. As a result, today in India, we have 609 testing labs, that is 431 public, 179 private laboratories are established in India across the country with the capacity of testing 1.4 lakh samples per day. And the capacity is being further raised to 2 lakhs samples per day. According to latest data from the Indian Council of Medical Research, that's called ICMR, currently 1.1 lakh samples are being tested each day in these labs. In this aspect, our university has been identified to establish a COVID testing lab, which is in the process of establishment. Therapeutic management for SARS-CoV-2 has largely been supportive, and to date, no specific therapy has been scientifically proven to reduce mortality. There is currently no vaccine to prevent coronavirus diseases 2019, that's called COVID-19, but the best way to prevent illness is to avoid being exposed to the virus. The virus thought to spread mainly from person to person and also between people who are in close contact with another within about 6 feet. This generally occurs through the respiratory droplets produced when an infected person coughs, sneezes, or talks. These droplets can land in the mouth or nose of a people who are nearby or possibly by inhaled into the lungs. Some recent studies have suggested that COVID-19 may spread by people who are not showing the symptom also. This is happening in India. Many times people say that we doesn't have the contact, but we have the coronavirus infection, the positive cases. How long will this pandemic last? So when we find a treatment for this vaccine, which drug should we give our patients? Although many unanswered questions, progress is being made and endless research continues worldwide to fight this pandemic situation. The only option available is to take care of yourself and people around you. Maintain at least 1 meter distance between you and people coughing or sneezing. Avoid touching your face. Cover your mouth and nose when coughing or sneezing. Stay home if you feel unwell. Refrain from smoking and other activities that weakens the lungs. Practice physical distancing by avoiding unnecessary travel and staying away from the large groups of people. I am very happy, I am very proud to inaugurate this webinar. So my pro-vice chancellor and other faculty members, they came and they requested me that they are going to hold this one. I immediately said yes, because this university should excel well and this department should develop also well. That's very important. My humble thing is that with all difficulty, with all this one travel, we got this department, and I hope that I wish this department should develop as one of the best center. So because not only the coronavirus, whatever diseases we don't know, this department should be strengthened and also should be capable of identifying what are the diseases which come, and also this department should help to find out the suitable medicines. So that is the research should go hand in hand, that is identifying the diseases and also to produce a vaccine or kind of a this one, the medicines is also very important. I hope this part of the country, particularly Gulbarga, the Kalburgi, is having the proud of this one. All media has announced the establishment of this one. I focus them. This was my intention to have and to develop this department as one of the best departments. I'm very happily inaugurating this one. My pro-vice chancellor struggled very much. Professor J.R. Naik, even though he's a biotechnologist, he was there heading the department in Gulbarga University, is having a very good laboratory. But his initiation and his help is very much is there to develop this one. I hope that my youngsters can do very good work and also to bring the law. With these few words, I am very happy to inaugurate this first webinar program of this topic is and scholars, one from foreign country and another from to very top institution in India speaking, and also I request them to should also conduct some workshops, the seminars, symposium, whatever we want. We always support this one. And with these few lines, the words, I am happily inaugurating this program. Thank you very much for providing me this opportunity. We say, I think it is an historic. This one is the program because we are starting this one and also developing the department. Thank you for providing me this opportunity. Thank you very much.
Now may I request our Pro-Vice Chancellor, also the Dean of School of Life Sciences, Professor G.R. Naik sir, to share his thoughts on the theme.
Well, good morning, our Honorable Vice Chancellor and the person behind many such webinars in the university and other activities, Professor H.M. Maheshwaraya, our Registrar, Professor Mushtaq Ahmed Patel, our Controller of Examination, Professor Kerur, the Coordinator of the Life Science Department, Dr. Durai Pandey, faculty members of the Life Science Departments who are here, faculty from History, the System Analyst and his staff, distinguished participants who are attending this particular very important webinar, our resource persons, Professor Barbara Broker, Dr. Sasidhar Manda, Dr. Muhammad Imtiaz Hassan. I welcome all of you. As rightly said by our Vice Chancellor, a historic day in the progress of Life Science Department of this university. Our Vice Chancellor has already told a lot of things about the department, faculty, as well as his ambition to make this department as a center of excellence on par like other departments which are present nearby, like University of Hyderabad or JNU. So I'm sure that this particular initiation will go a long way in objecting such type of things and achieving the target. When this department started, it started with a humble initiation to have a postgraduate degree in life science, and we had a very distinguished board of studies member, Professor Dayanand from Central University of Hyderabad, Professor Manjula Kumari from Bangalore University, and many reputed scientists have contributed. We had a team of experts under the chairmanship of Professor Geeta Bali, who suggested us that how the program should be there. Anyway, that is the part where the faculties contributed. When this was happening, there was an opportunity for the department to focus its presence, because suddenly the pandemic COVID-19 started, and that was the opportunity which we suggested the faculty to take it as a challenge and contribute something in this particular area, as well as for the Gulbarg area, which prominently needs such type of support. And as rightly uh, told by the Vice Chancellor, the MHRD, UGC, and ICMR are looking forward to establish a very sophisticated laboratory in, particularly now in the testing of COVID-19, but the same laboratory can take up many other such type of pandemic diseases like tuberculosis and all that, and it can become a center of such particular diagnosis, or as we discussed, or developing therapeutic proteins or therapeutic compounds which will be very helpful to the society. And in this particular direction, when we were looking, I'm happy that we have three faculties, particularly Dr. Asmita Samal, who herself worked in the medical biology at University of Medicine of Brisbane, Germany, and she had experience in working on Apollo Hospital, Hyderabad. She is with us. We have another faculty, Dr. Rakesh, who has experience in University of Hyderabad and molecular biology studies and experience in working in international institutes like Cuklifescience and other. We have Dr. Kavish, who has also a wide traveled experience in Canada and other countries. And so the faculty is well trained in the various molecular biology or recent trends which are there in the biology now. So this was the right opportunity for this faculty, along with the coordinator Dr. Durai Pandey, to look for such things and then take a immediate start. And in this particular direction, immediately a proposal is submitted now for ICMR and UGC, MHRD and state government to establish a state-of-art laboratory for diagnosis of these diseases, either molecular biology based or immunology based. And already university with its own grant, Vice Chancellor was magnanimous in giving around 40 lakh rupees. I'm glad to tell that RT-PCR has already come, debris has come, that old centrifuge has come, and many such requirements have come. With another month's time or so, the laboratory will at least start analyzing testing the samples for COVID, for which there is a demand here. And it will go a long way if that type of virology or infectious diseases center of excellence is established in this particular part of our country. With these few words, I thank Honorable Vice Chancellor for giving the encouragement by immediately providing the building and required infrastructure, the faculty who came forward immediately to prepare the proposal, and one of the step in that direction is today's webinar. I'm very grateful to Professor Barbara Broker from the University of Medicine, Brisbane, for accepting our invitation and preparing a talk on the management of this disease, and she is going to speak on her experience based on what is happening around the world. I'm also thankful to Dr. Sasidhar B. Manda, the Chief Scientific Officer, Apollo Hospital, Hyderabad, who is going to also tell about the management and what are recent trends in this particular disease which is around us, and everybody is worried about that. Dr. Muhammad Imtiaz Hassan, Assistant Professor, Jamia Millia Islamia, New Delhi, a very active research worker, is going to tell about how inhibitory molecules are being studied and what research is going on to restrict the growth of the virus and its impact. So we have a very good panel of people who are going to take care of the topic. I welcome all of them for this particular seminar. I am very glad that the faculty from Gulbarga Central University, Karnataka, and many other state universities, institutes from all over India and abroad, participants are present today to listen to all of these people. I welcome all of them, and I wish that this particular initiation by the Department of Life Science will go a long way to make this particular department as a center of excellence. Thank you for giving me the opportunity, and I wish again all the best.
Thank you, Professor Sir, for your encouraging words and opening remarks on the theme. Now it's time to welcome our first speaker of today's session. I feel privileged to introduce to you our guest of honor, Professor Barbara Broker. She is currently the head of immunology at the University of Medicine Greifswald in Germany. Her scientific interest focuses on infection immunology, especially multifaceted interplay between the Staphylococcus aureus and adaptive immune system. Having studied medicine and philosophy at the universities of Munster, Vienna, and Bristol, Barbara Broker has moved to London and for her postdoctoral studies in autoimmune diseases and become an immunologist. With her research group, she has contributed to several national and international research consortia. Since 2016, she is an elected member of Review Board, Microbiology, Virology, and Immunology of the prestigious German Research Foundation. We are grateful to Professor Barbara Broker for accepting our invitation to share her views. Although there was a time zone difference, we express our humble gratitude to her for sharing the presentation in recording, entitled Immune System and Behavior to Control SARS-CoV-2. A hearty welcome to you, ma'am, on behalf of Central University of Karnataka.
Good morning, everyone, and thank you very much, Rasmita, for this invitation to open your webinar on COVID-19. I feel honored, and I will just start with sharing my screen. So, as you can see, my name is Barbara Broker. I'm a professor of immunology at the University of Medicine of Greifswald, and this comes with a warning. I'm not a virologist, and I'm also not an epidemiologist, and so we are not even researching on viruses. But as you can see here, this is a culture plate with Staphylococcus aureus, and so my group is interested in the immune response and immune control of those bacteria. But I'm happy to share some thoughts about the immune system and the behavior in control of the coronavirus with you. And I hope that you may find that interesting. But let's first show you where I'm coming from and where our university is located. This is Germany, and there in the far northeast of Germany, there is an old town called Greifswald, and there is a very traditional university which was founded in 1456, and this is where we are located. Now, I don't need to introduce SARS-CoV-2 to you, which is known as coronavirus, or it is a coronavirus. It's the cause of COVID-19, this disease that is haunting the world. Most likely it's a virus of zoonotic origin, and as you know, the first cases were reported only in December 19, and now more than 6 months later, as aureus, the COVID-19 disease has become a real nightmare for the world population. These are the numbers as of yesterday. So there were more than 12 million of people infected worldwide. More than 500,000 died from that. In India, it's almost 800,000 infected with 22,000 people dying. And in Germany, we have diagnosed around 200,000 cases and almost 10,000 death cases. So how dangerous is it? The figures tell you a lot of it, but they don't tell you everything. And that is because testing and reporting policies very much differ between countries. And this very strongly influences the numbers. It influences in particular the numbers of unknown infections. And you know that many cases of COVID-19 go along with very few or even without any symptoms. And so it's very difficult to estimate the true mortality rate. And the estimates range from between 0.1, so that meaning 1 out of 1,000 infected, to 2%. And the death rate is very much influenced by age. So people older than 60 years stand a much higher risk of dying from COVID-19. And the older you get, the higher that risk. Then men are more at risk than women. And so in Germany, the fatal cases, 2/3 of the fatal cases are men and 1/3 women. Then it's comorbidities. And also the country and the healthcare system play a role. Of course, if the healthcare system is overwhelmed by COVID-19 patients, then their chances of survival decrease. But why has this virus become an emergency? I think one of the big problems is that it is very easily transmitted. The second, that the world population is immunologically naive. So there is no experience with this virus in the world. And it becomes more dangerous with increasing age. So that is a big problem in Germany with a quite old population. There is no vaccine and there is no causal treatment. Now, being an immunologist, I would like to focus on the problem of an immunologically naive population. And I will show you in a sort of didactic scheme what that means. Here you can see a naive organism infected by a dose of a virus, and it raises a primary immune response, as we say, and in this case, this does not save the animal. However, the adaptive immune system can learn, and we use that by vaccination. So if, for example, you take such a naive animal and you confront it with the antigen in an attenuated, in a less dangerous manner, then the animal will become immune. And if it is now infected with a lethal dose of the same virus, it raises a memory response. And this memory response is much stronger and more efficient, with a consequence that this immune animal has a much milder disease course, or even no symptoms at all, and it will survive. So the difference between a naive organism and an immune organism may be difference between life and death. Of course, it's not always so dramatic. And of course, the first confrontation with an organism need not be a vaccination. It can be a natural exposure. And now imagine COVID-19 or the SARS-CoV-2 being in the world for ages. This would mean that mainly young children become infected by the virus. And you know that young children usually have mild symptoms. They would then become immune, and COVID-19 may not become, would not be such a big problem. Now let's once again highlight the differences between a primary and a secondary, a primary and a memory response now in this more abstract way. So here you can see on the x-axis the day. Here you can see on the y-axis how can you say, the intensity of the immune response. And here you see in a confrontation of the immune system with an antigen A. And so until you notice the immune response of the adaptive immune system, a few days will pass. We call that the latent phase. And then you can see an immune response, which is rather transient, and then is reduced with control of the infection. And it will not return completely to baseline, but there will be immune memory. Now, upon the second confrontation with the same antigen, as you can see, the latency phase is much shorter. So the immune response kicks in faster, it's longer lasting, and it is much stronger. And so you could summarize the characteristics of a memory response with the Olympic terms Citius, Altius, Fortius. And that is what makes the difference. As you can see, this immune memory is specific. If you take a new antigen, the antigen B, the immune system will raise a primary immune response again. In view of all that, how can we control SARS-CoV-2? In the first instance, and the most important thing is to flatten the curve of infection, meaning to reduce transmission. And you are well aware of what that means. It means limiting contacts, keeping physical distance, and wearing face masks. Once the infection rate has decreased, then it will be possible to follow individual transmission courses, to detect and quarantine infected people to prevent further transmission. And that involves a lot of testing. So it's the term test, test, test. And of course, it is very important to provide good symptomatic medical care to those who experience a severe disease course. Now, Rasmita has asked me to tell you a little bit about the measures in Germany. And this is what I will do with the next few slides. Here you can see on the black background, you can see the diagnosed cases. And you can see this is beginning of March. And so you can see it is gradually starting to increase. And then on the 11th of March, the WHO declared COVID-19 a pandemic. And on that very day, the Chancellor Merkel addressed the Germans and said, this is serious. We have to take measures. We have to be careful and flatten that curve, because otherwise, with an exponential increase of infections, there will be the cases will overwhelm the healthcare system. And then things started. So on the next day, the hospitals were told to stop routine treatments to make space on the intensive care units for COVID-19 cases. One day later, nurseries, schools, and universities closed. And then a couple of days later, the shops were closed except for those selling the daily necessities. But as you can see, the infection rates were still increasing. And so a week later, Germany went into lockdown. Lockdown was rather mild in my country. So there were contact restrictions. Private meetings of more than 2 households were basically forbidden. Especially it was mentioned that kids should stop visiting their grandparents because they are in the high risk group. And in public, so we were always allowed to go out, to walk, to go to the supermarkets, for example, but also to walk, to jog, to do sports, but no more than 2 people from different households together. And we were also advised to keep a distance of more than 1.5 meters. And interestingly, polls reported that 99% of Germans agreed with those measures. What does that mean? Here you can have a look at the Berlin-Brandenburg Gate, and this is during the World Football Championship, and this is during the lockdown. And then we waited, and we were staring at the numbers, and it was quite a painful period because it took 3 weeks to be really sure that the lockdown was effective. Fortunately, it was. And it took still more time until face masks were recommended, or they were even made mandatory in shops, in public buildings, and in public transport. And this is because at the beginning it was not clear whether they would protect it all. There was, of course, a huge demand of face masks, of professional face masks in the healthcare system. And face masks in public are not in the German tradition. And so then in end of April, they were then made mandatory. And 7 weeks then after the lockdown, it was clear that the numbers were going down and the contact restrictions are now gradually released and eased. Now the control of COVID-19 in Germany is on a more regional basis, and there is a new rule called 50 out of 100,000 over 7 days, and I will show you what that means in a moment. And now schools are gradually opening. Right now, however, there are summer vacation, but after the summer vacation, all children will go back to school. So this is the 50, 100,000, 7 rule. Health care or health authorities are organized in districts in Germany. So the district health authorities monitor the cases, they trace the transmissions, they identify cluster super spreading events, and they enforce quarantine. So the rule is following. If over a period of a week of 7 days, there are more new infections than 50 per 100,000 population, then additional measures are taken. Otherwise, the health authorities will identify infected people. Those will be quarantined at home for 14 days, and their contacts will be traced and also quarantined and tested. And so that is how we hope to further control the spread of the virus. And as you can see, fortunately, this appears to work quite well. So that in June, it was then decided to reopen the European borders. And a few days later, there was also a Corona app to ease the contact tracing. So we all hope that we will be able to live with such very low infection rates of Corona until we have better means of control. And wherever in Europe such measures have been taken, it turned out that they work. And so I always tell the people in my group and our students, give an example, keep your distance, and use a face mask. And of course, especially in the hospital where we are working, we are using face masks throughout. So this will slow down SARS-CoV-2. But how can we stop the problem? You know that. We are all waiting for vaccines. And active vaccines, I think, are the most promising way to really make a difference. Then I will briefly discuss passive vaccines. I will not discuss antiviral drugs or host-based therapies. Of course, intensive care is important in severe disease courses. And in the UK, a large clinical study has revealed that the cytokine storm, the massive inflammation that goes along with COVID-19 in a subgroup of patients can be mitigated using corticosteroids. So let's turn to vaccines. Active vaccination. I've shown that to you before. If you confront a naive organism with antigens, the organism will react to that. It's the adaptive immune system and develop immune memory. Upon a secondary confrontation with this antigen, there will be an immune memory response, which will be faster, longer lasting, and stronger, and also of different quality. So it will be stronger, of different quantity, it will be of different and more appropriate quality in the most cases, and it will be specific for the antigen the organism has been primed with. And everything going well, this will then lead to protection from the challenge with an infectious agent, in this case, SARS-CoV-2. So is there a chance for us to develop an active vaccine? I once again stress that vaccination relies on immune memory, on antibodies, B cells and T cells, and I think there are indications that it will be possible, that that should be possible. And these are the arguments. Infections with SARS-CoV-2 are usually cleared within 14 days or even less than that. No chronic infections have been reported, and no viable virus is retrieved from the throat after 7 days of infections. We know that it sometimes goes into the lower airways, but also there it will be cleared. There are no reinfections, or maybe very, very few. And animal data also suggest that vaccination can induce protective immunity. So the chances are good. Because we know that the adaptive immune system is able to clear SARS-CoV-2 and protect from later infection. However, right now we do not know how long that natural immunity lasts and how long it will protect. And this also, of course, impacts on the vaccine trials and on the vaccine anticipated vaccine schedules. So how can you vaccinate against SARS-CoV-2? There are about 130 vaccines in development, and I'm sure some also in your country, because in your country there are the huge vaccine producing facilities of the world. Most of those vaccines target this red spike protein, which is the viral attachment protein. And if antibodies are raised against the particular epitopes on the spike proteins, they can prevent virus attachment to its receptor, which is the angiotensin converting enzyme 2. Sometimes it appears that harmful anti-spike antibodies may be generated, which are not neutralizing, but rather disease enhancing. But it appears to be a relatively moderate problem, and let's hope that this is correct. T cells are, of course, also important for combating viruses. So what are the strategies? The first and fastest are RNA-based vaccines. These are relatively easy to generate because it is easy to synthesize nucleic acids. So the principle is to synthesize mRNA, and they're coding a viral protein, the spike protein, and then encapsulate it with lipid nanoparticles, which then merge with host cells. Those cells then will translate this RNA into protein, and that protein will then trigger an immune response, namely an immune response of T lymphocytes and B cells, which then in turn will make antibodies. So this is straightforward, and there are numerous companies who are following this approach. Until now, there is no working example. So COVID-19 would be the first. The second, a very elegant way, are the viral vector-based vaccines. So here, there are already so-called vector platforms. Those are harmless viruses. For example, adenovirus, chimpanzee adenovirus, cytomegalovirus, or measles virus variants. So they are attenuated variants with very low pathogenicity. And those viruses can be made to take up novel genes that encode the antigens of other viruses. And so the idea is to take one such virus, let's say, for example, this adenovirus here, and transduce it with SARS-CoV-2 antigen encoding genes. And then if you then vaccinate an individual with this virus will then be synthesized in the host cells, by the host cells, and it will have on its surface SARS-CoV-2 spike protein, and hence it will raise an immune response against SARS-CoV-2. So adenovirus 5 is being used in the Chinese clinical study, and a modified chimpanzee adenovirus in studies that are performed by Oxford University. There is a working example here, and that is an adenovirus encoding Ebola antigens. The classical way of making a vaccine, a component vaccine, would be protein-based vaccines. They are more laborious, but they are tried and tested. So the idea is here to clone and express a recombinant antigen in mammalian cells or in plant cells, and then purify that protein, add some adjuvants in many cases, and then use that as a vaccine. So there are many working examples, for example, hepatitis B, some influenza vaccines, and also human papilloma virus vaccine. So these are the main principles for an active vaccine against COVID-19, against the SARS-CoV-2 virus, and let's hope that some of them will work. In addition, we should also consider passive vaccination. Passive vaccination means that we will use the sort of ready-made memory response of an immune system to transfer those moieties to a naive or severely ill patient, and so to provide that patient with ready-made immune memory. And so this is the principle to transfer virus neutralizing antibodies into patients. The fastest approach is to obtain hyperimmune serum from convalescent COVID-19 patients, and that's being done in Germany, for example, but all over the world. From this serum, you could purify specific antibodies and use them to help people with severe COVID-19. There are working examples. One of them, the most commonly used probably, are anti-tetanus toxin antibodies that are applied to people who are at risk with an uncertain vaccination status. The technically more advanced way would be not to purify the antibodies from human sera, but to generate monoclonal antibodies that can neutralize SARS-CoV-2. You do that, I will just briefly describe that to you. You need to obtain human peripheral blood mononuclear cells, human immune cells, as a source of SARS-CoV-2 specific B cells. So you will probably get them from a patient during the disease course or from a reconvalescent individual. You can then take those B cells, select them, clone the antibody coding genes, transduce them into an expression system, and which then will generate those antibodies. There are, of course, numerous monoclonal antibodies approved for therapy, for cancer therapy, for autoimmune disease mainly, but there are also two in Germany, that are approved for therapy of infectious diseases. And one is the anti-RS virus monoclonal antibody pavilizumab, and another one that is will neutralize Clostridium difficile toxin, bexlotanil. So this is possible, but it will take time. Let me take the last 3 minutes to introduce a very new concept to you, and that is trained immunity. Trained immunity is a sort of memory which is different from the memory that I have described to you before. Before, I was stressing that immune memory was a core competence of the adaptive immune system. We call that immunity or immune memory. However, the innate immune system can also develop some sort of immunity, and this is known as trained immunity. And I will show you how that was discovered. We all know that tuberculosis and measles are big killers, and that vaccines against those pathogens save lives. Interestingly, fascinatingly, The BCG vaccine and also the measles vaccine saved many more kids than would have been killed by the pathogens. So we conclude that vaccines can have beneficial effects that reach beyond that specific pathogen target. And this is known as heterologous vaccine effects, or non-specific or off-target vaccine effects. And so the question was raised whether tuberculosis vaccines can strengthen the immune response against SARS-CoV-2. How to test that? Well, this is going to be tested. I will show you that in a moment by testing additional parameters within ongoing clinical trials. But before showing you that, I will just once again show you an experiment that clearly demonstrates heterologous protection. And this is the study design. So people were vaccinated with BCG, and there was also a control group. And then those people were infected with a virus, with yellow fever virus, but with an attenuated yellow fever virus, because this infection was a vaccine. So they were vaccinated with yellow fever virus, which is a very, very efficient and very good vaccine. And then viremia was monitored. And as you can see, those people who had been BCG vaccinated, they had a much lower viremia than those who had not been BCG vaccinated. And this difference was associated with higher levels of interleukin-1 production and epigenetic changes in the monocytes. So while BCG and yellow fever virus do not share any antigens, the BCG vaccination could still train the immune system to be more efficient when confronted with this virus. And hopes are that this may also be the case with coronavirus. So there are several ongoing studies. One is using BCG versus a placebo. This is, of course, a tuberculosis study. But now people can check whether it also influences infections with SARS-CoV-2. And here you can see that there is another experimental or pre-experimental vaccine manufactured in India. This is VPM 1002. It's a BCG variant, which protects much better, and there is now a phase 3 clinical trial planned. And in this case, besides tuberculosis, which would be then the specific effect of this vaccine, the impact on SARS-CoV-2 infections can also be monitored, meaning that they will now also monitor the heterologous vaccine effects. And I'm very optimistic that that many of those approaches will result in vaccine strategies that will be helpful. They will not eradicate SARS-CoV-2 from the face of the earth. I don't think that this will happen, but I'm very positive and hopeful that they will alleviate the situation of this pandemic. So thank you very much for your attention, and stay healthy.
Thank you, Professor Barbara, for giving a comprehensive overview on the SARS-CoV-2 and sharing the various strategies that have been implemented in a global context. So thank you very much for this wonderful presentation. Before moving on, I would like to mention that any questions addressed to Professor Barbara can be communicated to us at lifesciences@cut.ac.in, and we will revert back with the response via email. So now we move on to our next speaker. It gives me immense pleasure to introduce our next speaker of this day, Dr. Shashidhar Manna. He is currently the Chief Scientific Officer at Apollo Hospital Educational Research Foundation. He is also the founder of Urcogel Bio, a biotech startup engaged in development of exosome diagnostics for oncology and neurodegenerative has pioneered the exosome biology platform at HERF and expanded the product portfolio into various applications such as diagnostic, drug delivery, exosome engineering, and therapeutics. He has received his PhD from Heinrich Heine University at Düsseldorf in Germany and was a postdoctoral fellow at University of California. His research areas involve inflammation, drug repurposing, genomics, proteomics in multiple sclerosis and lupus. With over 15 years of research experience, he has co-authored a number of international publication case studies. Thank you, Dr. Shashidhar, for accepting the invitation. In this talk today, he will discuss on recent developments in COVID disease management. Central University of Karnataka heartily welcomes you, Dr. Shashidhar.
Thank you, Dr. Rashmita, for giving me the opportunity to. This will be the overview of my presentation here. So I will be making few slides about the biomarkers overview, and then I will be sharing some slides regarding the COVID-19 pathology and the major characteristics of the COVID-19 in terms of the cytokine storm, the multi-organ damage, why COVID is so infective, and finally I will end up with the research overview of COVID-19. So basically, coronaviruses are a large family of viruses that cause from various diseases from common cold to severe diseases including MERS and SARS. So the most recently discovered coronavirus is the COVID-19. So it started in December 2019 as a cluster of atypical pneumonia patients linked to Wuhan district in China. A novel coronavirus is discovered is discovered. So the family of the coronaviruses in the recent history is in SARS-CoV-1, which is from 2002 to 2004, followed by MERS-CoV virus, which is currently ongoing still, and the SARS-CoV-2, which is the recent pandemic or the global emergency that has been declared around January. So this slide gives you an overview of the history of the pandemics, right from the Androinian plague to the current novel coronavirus. So the 2019 coronavirus is a severe acute respiratory syndrome, while the disease associated with it is referred to as the COVID-19. So the COVID-19 is a novel coronavirus of zoonotic origin, and it belongs to a family of beta coronaviruses. So it is a single-stranded virus, and it has the typical crown-like appearances under the electron microscope. That is the reason why it is known as the COVID thing. So the typical proteins that are present in the around the COVID-19, which majorly includes the spike glycoprotein, the envelope protein, and the matrix glycoprotein. So the spike microprotein, the glycoprotein, is responsible for the receptor binding, and the envelope protein here is responsible for the virus infectivity. So the matrix glycoprotein here actually interacts with the envelope protein to form the viral envelope. The spike protein of the coronavirus is 10 to 20 times more likely to bind to the human cells, which could be one of the reasons why the infectivity of COVID virus is so high. So this slide shows you the impact of the COVID-19 globally, and this shows you the total new cases and the confirmed cases, which almost cross 14 lakhs all over the world. And coming to the current knowledge of COVID-19, like the source and reservoir, bats, and maybe there is an intermediate host. And the mode of the transmission is droplet or contact, and recently aerosol infection has been also included. So the incubation of this COVID-19 is around 5 days, 2 to 14 days on an average. And the capacity of human to human transmission is sustained everywhere, and the period of communicability is around 14 days. So why is the major reason of the spread is the viral shedding, which is the regarded as the major reason for the spreading of the COVID. So this slide actually shows you the different types of other colds and flus and how COVID-19 commonly overlaps with the cold and the flu, like in terms of the cough, the diarrhea, the fatigue, the fever, the runny nose. So there is a significant overlap between the cold and the flu, which is also one of the major alarming reason here. So the second thing is the COVID-19 disease severity. So the COVID-19 typically follows three patterns. One is the asymptomatic, which is the mild infection. 80 to 81% suffer from the mild infections with flu-like symptoms, and these people can be recovered at home actually. And 13.8% are the severe pneumonia patients. Uh, they typically encounter the shortness of the breath, and almost 4.7 to 4.8 are the critical patients which finally end up in respiratory failure, the septic shock, and also the multiple organ failure. So coming to how actually the COVID-19 is managed in the labs, managed in the hospitals, there are three layers of complexities here. One is primarily the diagnosis, the molecular testing. So currently nucleic acid amplification, that's RT-PCR, is the typical gold standard, and you have the samples from the upper and the lower respiratory tract. And then you have the serology testing, which is generally done through either the lateral flow or the ELISAs, where you measure the whole body temperature and the specimen samples. So anticipated here of screening, the identification of the convalescent donors, and assessment of the immunity. The next thing is the biochemical monitoring. Biochemical monitoring includes markers of inflammatory, cardiac, hepatic, and renal. So the one reason why COVID-19 is actually affecting multiple uh, reasons is due to its infectivity with the ACE2 receptor, which I will be discussing in the next slides. So in this slide, you are primarily seeing what are the typical COVID-19 diagnostics. One is the nucleic acid testing, where you would be amplifying the envelope protein and all the RDRT, that is the RNA dependent RNA polymerase genes. So false notificated lab setup are some of the limitations of the nucleic acid test. And the step is testing for the OM and to ally the CLA. So discrimination among the negative infections is typically when the RT-PCR is negative. That is when test is used. So coming to the clinical pathology of COVID-19. So nose and throat are the established routes for the viral entry. And ACE2, that is the angiotensin converting enzyme, is a transmembrane protein, a peptidase actually, which facilitates the viral entry into the body. So ACE2 degrades the angiotensin to angiotensin 1 to 7. So that's a vasodilator, which is how a major impact is being done on the cardiovascular pathways. So there is a debate whether the ACE2 inhibitors have to be stopped during COVID-19 infection, which currently is subsided. So in this slide, actually you see that there are typically two phases. One is the viral response. Like after the infection is done, there is typically the viral response, and then is actually the post in the immune response. So, and the common symptoms of the COVID-19 are the fever, the dry cough. This according to the majority of the infections, and the fatigue, sputum, the sore throats, diarrhea, and so on. So before going further, like what are the typical biomarkers that are currently being used? Uh, so these are some of the properties of an ideal biomarker. That the biomarker should be detectable prior to the organ damage. It should be highly sensitive and specific for the disease of interest. It should correlate with the disease severity, and it is easily accessible, and more better is the non-invasive nature, which is the blood, urine, tears, and the saliva. And the biomarker should be analytically stable, and it should be reproducible and cheap to measure. And the biomarker should indicate a specific treatment mechanism. So typically in COVID-19, what are the typical biomarkers? So here in this slide, you are actually seeing the asymptomatic, the mild, moderate, the severe, and the critical phases as discussed in the viral phase and the host response to the viral infection. So during the asymptomatic, which can be maintained in either home or the community, you can see that the normal lab test, uh, routine lab test can be used. That's the mild lymphopenia, finally end up in the lymphopenia. And in the mild moderate thing, you actually see an increase of the prothrombin time and an increase of the ferritin and the dimers and the LDH and the hypoxia, which is typically more than 92, and the severe cases, which is typically treated in the hospitals. There is an increase in the D-dimers, an increase in ferritin, transaminases, and there is a decrease of the hypoxia, which is less than 92, and an increase of interleukin 6 and the CRPs, which finally in for 5%, typically in 5% of the patients typically end up in the intensive care units, which there is a marked elevation of the inflammatory biomarkers. So currently, what are the biomarkers that are being currently used in the heart? These are the troponins, the neotropic peptides, and to measure the coagulations of the D-dimers, to measure the inflammations, that is the procalcitonin, the C-reactive protein and the SAS, and the kidneys is the cystatin, and muscles and the myoglobin. So COVID-19, typically there is organ damage all over from the brain to the kidney in terms of, and in the lung there is an inflamed and damaged alveolus, and in the GST tract which typically ends in diarrhea. In the eyes you have the conjunctivitis and the blood clots in the case of the inflammatory disease and the renal damage. So you can see that COVID-19 damage is typically uh, pleiotropic. It's entirely over the range. So what could be the main reason there? So there are some of the important phenomena that have been recently that has been cytokine storm. So in order to briefly explain what a cytokine storm is, it's a hyperactive immune response which is typically characterized by the release of interferons, leukins, chemokines, and some several other factors. And you can see in the slide, in an uninfected individual, you have the typical the CD4 and the CD8 cells where there are no symptoms. And in the moderate COVID-19 patients, you have a damaged epithelium due to the SARS-CoV infection, where the inflamed macrophages release the TNF alpha, the interleukin 6, and the interleukin 10. And in response, there is an interferon gamma expressing CD4 T cells, which finally results in fever, cough, and fatigue. And you have the severe mode, which is actually results in fever and cough, and there is an upregulation of interleukin 6 and LDH, and there is a downregulation of lymphocytopenia. And there is the COVID-19 in what happens in the renal failure. In renal failure, everything happens due to the presence of the ACE2 receptors, even in the kidney. There is an elevated serum creatinine, elevated blood urea nitrogen, and there is a decreased glomerular filtration rate, and there's a proteinuria and hematuria, finally leading in acute kidney disease. So renal disease is also the important complication which clinicians should consider. The next spectrum is the cardiovascular disease. So the spectrum ranges from the acute coronary syndrome to arrhythmias to heart failure, finally to stress cardiomyopathy, cytokine dysregulation, and myocarditis. A important mechanism we should be aware during the COVID-19 is a typical coagulopathy, which is known as the disseminated intravesicular coagulopathy, in which there is an upregulation of D-dimers, fibrinogen, prothrombin time, and the platelets. So here you can see when there's too much clotting, you have an organ damage, and too little clotting, you have there is an use of platelets and the clotting factors here. Uh, COVID-19. So, what are the differences between the survivors and the non-survivors? So, this Lancet paper shows you that the differences between the survivors and the non-survivors typically comes after the 9th and the 10th days. So, here in the survivor population, you have the sepsis and the ARDS, and typically they are discharged around the 20-21 day. The course of the infection goes away. But in the non-survivor, you can see that ARDS typically comes at the 12th day. There are variations, but around the 14th and the 15th day, you have the cardiac injury, the renal injury, and there is a secondary infection which leads to death. So, what are the biomarkers that are helpful for managing COVID? So, as discussed previously, is the coagulopathy, and the most important are the D dimers, the platelet count, and the prothrombin time. So you have the three different stages. That's the stage 1, stage 2, and the stage 3. And you can see that there is a variation of the D dimers. There is a 2 to 3 fold increase, 3 to 6 fold increase. This graded in this normal, and the second stage is 100 to 150 10 cells per liter, and its platelet count is less than 100. Prothrombin time in that stage 1, which is managed in the ward, is normal. It's minimally prolonged, and it's typically prolonged in the stage 2 or the serious thing. So coming to the different biomarkers, the cardiac biomarkers what are commonly used are the high sensitive cardiac troponin in the hospitals. So it is reported that this high sensitive troponin is elevated in COVID-19, and it's more elevated in the milder forms. And so also myocarditis, and microarrhythmia is also resulted in the cardiac biomarkers. And then is the natriuretic peptide response in COVID-19. So NPs are biomarkers of myocardial stress, and they're typically elevated. And also additional biomarkers like the brain natriuretic peptide, which are associated with the unfavorable ARDS scores. And values greater than 88.64 picograms have more risk of hospital stratification, and BNP levels greater than 100 picograms are likely to develop severe pneumonia. So, what this literally means that is these biomarkers are useful in personalizing treatment and able to predict how a person will move, and greater care can be given to those individuals. This is some of the data of our hospitals, where typically you can see there is an of a hospital which are being treated for the COVID infections. So here you can see there is a down regulation of lymphocytes, and which finally ends in lymphopenia and monocytes, the neutrophils. This is how the neutrophils increase is happening in the platelets and the basophils. Um, And from the lymphocyte ratio, the platelet ratio, and the monocyte ratio, you can actually calculate the different ratios, that is the NLR, the platelet lymphocyte ratio, the monocyte to lymphocyte ratio. And typically these ratios are variable and vary in during the infectious diseases. And so this is the typical ROC curve of the different NLR, the LMR, and the PLR CRP, which can be used as a predictive value, as a prognostic factor, how the course of the disease is likely to be in a patient. And these are the hematological ratios of the COVID-19 patients in our hospital. These are the ROC curves calculated from the calculated from the lymphocyte ratios. And typically the NLR is the neutrophil to lymphocyte ratio. So here you have an area under the curve of 0.824, and the lymphocyte LMR, which is 9.59, and PLR, which is typically 0.946. This is the combined ROC curve of all the different ratios. And the CVB profile of the COVID-19 patients, of the COVID patients, typically you can see how there's a decrease in the hemoglobin, the hematocrit values, Then there is increase of MCHC, the total leukocyte counts of the COVID patients. So now we have an idea of about the CVP, the profile. Now let us just go about the different inflammatory biomarkers that are used in the COVID-19 management. The primary thing is the C-reactive protein, the procalcitonin, and the serum amyloid protein. So C-reactive protein in normal viral infections isn't upregulated, but in COVID-19 it can be used as a, along with the CBP, can be used in triaging of the inflammation. So why inflammatory biomarkers are used in COVID is because COVID-19 is not a simple respiratory disease, it's an hyperinflammatory disease infection. Serial PCT measurements could predict severe form of a disease. So all these biomarkers can be used in COVID-19 infection and also may be used to correlate to secondary bacterial infections. And another important inflammatory biomarker is the D-dimer levels. So D-dimer is a fibrin degradation product which is typically formed when the blood product is degraded during the fibrinolysis. So D-dimers values are upregulated in 36 to 43% of the COVID-19 patients, and D-dimers helps in adjunctive use of antithrombotic therapies which are currently being used, and you can see the stark difference between the survivor and the non-survivor population, how the D-dimer values are so this is the data of inflammation profile of the COVID-19 patients. Typically, you can see there's a surge of interleukin 6, D-dimers, the LDH, that's the lactate dehydrogenase, the CRP, and the procalcitonin in the COVID-19 patients in our facility. And during the renal, and also during the renal disease, we can see from the Kidney International Journal here. So you can see that as the proteinuria, the hematuria, the baseline creatinine and the C creatinine and the acute kidney injury increases when there is a typical risk increase of the values. And again, this is our COVID-19 laboratory profile of the renal profile. Here you can see that the pre-albumin serum, the albumin to globulin ratio, there is an increase of the urea serum in case of COVID patients, an increase of urea creatinine. So typically this shows that there is a renal mismatch. There's a renal mismatch in case of the COVID patients, and also there is an imbalance in the electrolytic profile in case of the potassium, the sodium, and the phosphorus in the COVID patients here. So this is a summary slide which gives you what's happening in the hematological thing. There's a downregulation of lymphocytes, upregulation of neutrophils, there's upregulation of CRP, PCT, ferritin in the interleukin 6. So in upregulation of D-dimers, the prothrombin time and the fibrinogen, and the electrolytes, there is a decrease in potassium, sodium, and calcium. And the hepatic, there is an upregulation of the L9 AAT, the total bilirubin, and the renal, there is an upregulation of creatinine, the urea. So all these along can be used in the therapeutic management of the COVID-19 infection. There is a new finding that COVID-19 also increases the risk of diabetes. So there are many mechanisms probably that how this could happen. Probably one mechanism could be the cytokine storm, that is typically the interleukin 6, and there is destruction of the pancreatic beta cells, and also there is a drug to drug interaction probably due to the steroids that could be probably being used. So there, and this is our hospital data, like what's happening with there's an upshoot of the interleukin 6, and there's an upregulation of LDH and the serum creatinine, and we see that there is an upregulation of glucose. So we didn't demarcate between the diabetes and the normal thing, but as an average you can see that most of the patients have a surge of glucose, which is a matter of interest that is being investigated here. Also during COVID-19, there is neurologic complications associated, typically due to the movement, the migration of the migration of the virus through the olfactory epithelium to the brain, and also through the monocyte macrophage activation, which passes the blood-brain barrier, and also through the peripheral nerves. So these are the different mechanisms which there could be a direct effect of the COVID-19 on the brain, and there could be an indirect effect. Uh, Indirect effect. So what are the major CNS complications? One is the GBS syndrome, the cerebral vascular disease. So the direct vascular infection, the para-infectious, and the vascular thing. So these are the time frames where uh, how this can be diagnosed. So currently, these are the different knowledge gaps in the COVID-19. Is the limited knowledge here in case of the asymptomatic disease component, the epidemiology, the knowledge of the mutants of the COVID-19, and the transmissibility issues that, and further is the post-recovery implications in terms of the cerebral stroke, the neurological complications. So as more and more research is added up towards the COVID-19, much data is being compiled in this area, and we in our research foundation are also working in development of COVID-19 based applications. So we are trying to develop saliva and urine based exosome diagnostics. We are trying to, so we generate huge amount of imaging data. We are trying to develop an AI approach towards it, um, using the clinical thing. So we also have a clinical trial division which is now testing new repurposed drugs for COVID-19, and we are also working on identifying new actionable factors in convalescent plasma, because the convalescent plasma is highly variable. So we are trying to explore new factors which could partly explain the variations in the convalescent plasma, which is a promising COVID-19 option. So I just briefly summarize you what we have gone through. So biomarkers have a very important role in predicting the course, the progression of the COVID-19, and the risk actually increases with different comorbidities, which is diabetes, hypertension, cardiovascular diseases, cancer, chronic liver disease. And a subset of COVID-19 patients express the cytokine storm, which finally results in renal hypertension, neurological complications. Uh, COVID-19 is associated with multiple cardiovascular, diabetes, renal, and neurological complications. So, developing algorithms from the laboratory data can be used in personalizing the treatment and predicting which patient will actually move to a severe course, and proper management can be given by using the data from the thing. Uh, so, thank you, Dr. Rashmita. So,
Thank you, Dr. Sachibai, for this nice presentation. I would like to ask a question related. So, there are a lot of talks on airborne diseases, and like COVID can spread to airborne more. Can you comment something on this?
yeah, I think WHO recently upgraded the risk level towards the aerosol contamination, because it can be expected that as COVID-19 is primarily a nasal and a saliva throat based infection. It's the primary place where it gets lodged, and the high risk probably due to the COVID-19 infectivity probably could be the aerosol contamination. And I think proper usage of the mask and decontamination of the place could definitely bring proper um, control of the COVID-19.
Okay, so we have one more question. Can influenza biomarker be used for treatment of COVID-19?
So as you can see, that typically COVID-19 is an inflammation-based disease and hyperinflammation-based disease, and affecting different organs because of the pleiotropic effect of ACE2 in almost all the different organs. Yes, influenza-based biomarkers in terms of the CBP profile, the inflammation markers can be used, and specifically when there are different organs that are in terms of the COVID-19, the different organs that are involved can be used with different the different biomarkers can be used.
Thank you, Sushila. There is one more question popping up, but it's like a comment, I would say, from IIT Delhi, Jatin Kumar Sharma. So he says that they have developed cost-effective and pure indigenous corona test kit, but still we are using imported kits. Why are we not able to use the IIT Delhi kit yet? Can you comment anything on this?
So I'm not exactly aware of this IIT kit, but as far as I can say that if the kit has been validated with different other kits with the golden standards that are available in the market, and whether they have applied for the DCGI, there is a rule of the land where the kit has to be established, standardized, validated, and probably if everything has been done, I don't see any reason why people are not using it.
That's quite inspirational. Thank you very much, Sashidhar. Now we move on to our next speaker, the last speaker of today's session, Dr. Mohammad Imtiaz Hassan. He is an assistant professor of structural biology in the Center for Interdisciplinary Research in Basic Science, Jamia Millia Islamia Central University at New Delhi. His research interest includes drug design and discovery, structural biology, and SARS-CoV-2. He is an outstanding researcher involved in many drug discovery projects with great social and industrial impact. I feel proud to announce that he is among few scientists in the country who published a tremendous volume of high-quality publications, over 260, in a very short time of about a decade. He is globally recognized for his excellent research in the area of drug discovery, which is evident as he has been elected as a Fellow of Royal Society of Chemistry in UK, as well as Fellow of Royal Society of Biology in UK. Currently, his lab design and develop potential drug candidates to address clinical management.
Good afternoon, everyone. I am pleased to know to deliver my talk on the current emerging topic on the SARS coronavirus. And I am much thankful to Dr. Rashmita Samal for inviting me and giving the opportunity to speak in the great audience. So today I'm going to talk about, in the last two previous lecture you have listened about more about the immunological component and the clinical prospects of the coronavirus. But I am going to talk about the different thing, that is the structure-based drug design and discovery against the coronaviruses. Because if you see the major two component of the addressing the COVID challenge is, one is the at the vaccine level and another is at the level of the drug discovery. So both are the way we are trying to fight and address this global pandemic, and many people are getting success, whether it is major or minor, but at all the level, people are trying hard across the world to. So in the last presentation, you already heard about the coronaviruses. It is one of the member of coronavirus family, and if you see structurally or at the sequence level, it is much closer to the last two viruses, that is the called coronavirus 1 generation, simply called SARS coronavirus, that emerged in 2002, and the MERS coronavirus, that Middle East Respiratory Syndrome coronavirus, that emerged in 2013. Despite it having a high similarity to the bat coronavirus, and people are based on that sequence similarity, it have a notion that it is emerged from the bat. So if you see, the severity of disease is actually depending upon the, of course, pathogenicity, but after the pathogenesis, there is a one phenomena called cytokine storm. That is the release of cytokine, and that is associated with the high fever and the clinical symptom. That are the major problem with this coronavirus infection. And people are trying from the different ways, different angle to address this coronavirus disease by targeting SARS cytokine storm, targeting infection, targeting different aspects of the pathogenesis. And many of the already available drugs for the different viruses are trying in clinical practice, including the chloroquine and hydroxyquinone, that all the drug given some beneficial impact, but so far none of the drug is, you can say that it is purely cure this disease. So let's start with the structure of coronavirus, and this is the simple structural presentation of the coronavirus. You can see it contains four different proteins, four different structural proteins, and these structural proteins plays a significant role. The first structural protein you can see here, it is the spike protein, and spike protein is the major protein which actually starts the pathogenesis process. The spike protein actually attaches the virus to the host surface receptor, and then through which the infection process starts. Another interesting protein is the nucleoprotein that synthesizes nucleocapsid of the virus and in viral RNA formation, and the ribonucleoprotein. Another important protein is the M protein, that's called membrane protein, that synthesizes viral membrane assembly for the mature viral particles. And lastly, this is the E protein, that's called the envelope protein. It is a part of the envelope and it having role in the assembly of the virions. So these all four proteins are major structural proteins, and that plays important role in the viral assembly, multiplication, and pathogenesis. It is very important to know that these four proteins are sharing high sequence similarity among all the viruses. And these proteins are playing very significant role in the multiplication of virus and the life cycle of virus. That's why these proteins are targeted by different groups for the drug design discovery as well as for the process of vaccine development. Just I'm showing you a brief outline of the viral cycle, for especially for the graduate student that could know that how this virus proceeds and what are the places where we can target, and how the actually for the drug targeting, how we explore the pathways of the viral infection. You can see this is the viral particle. It attaches themselves to the angiotensin converting enzyme 2 receptor, and through binding through this, that enters to the host cell, and then they release their RNA, because it is the RNA virus which integrate to the host virus, and then it sorry. So after that, it start to synthesize their own proteins, and then forming the new viral particle and released out. If you see the during the process of this life cycle, there are several inflammatory cytokines are released, like the interleukin beta, TNF alpha, and then through the Th17 activation. And these cytokines are actually responsible for the cytokine storm, and that is the associated with the fever. Gene sequence of the sound, it is having about 30,000 variants, and 12 expressed proteins are there, and the genetic makeup of this virus is closely resembles to the SARS coronavirus as well as the MERS coronavirus. So that's what the people are suspecting, that whatever the etiology, what are the molecular basis of pathogens that are sharing high similarity to the MERS coronavirus and the SARS coronavirus. That's what the whatever the drugs and whatever the knowledge previously scientists gained from the earlier virus, they are trying hard to imply with the this one. So I just compared the sequence of all the three genome, and you can find there that their sequence are almost equal in length, although MERS coronavirus sequence is little longer, and all having the four structural protein. However, the MERS coronavirus having extra non-structural protein, and these are the open reading frames and their arrangement in the different viruses. So what we first did, we analyzed the sequence of SARS coronavirus genome, and especially each non-structural protein, because people are assumption that the non-structural protein, since it is don't have the function, it may not have the since don't have a structure, so it may not have the function. But we try to do the extensive sequence analysis, and we deciphered the first we deciphered the tendency to forming the structure, and then we did the gene ontology analysis, and through that we found that these are the different function attributed to the all the different non-structural proteins. In the table, I showed all the function of these like NSP1, NSP2, NSP3, and accordingly, these are the having the different function. So after annotating this function, we established that the role that which genes are highly important in terms of pathogenesis, in terms of viral survival, and all these things. And this paper recently we published in the BBA. After that, we perform the analysis of all the structural protein, or you can say that all the target proteins, which are the target proteins of the virus. So spike protein is one of the major target, especially target in terms of the antibody generation or the vaccine development, because it starts the process of the pathogenesis. And you can see this is the 1272 residue long polypeptide having 3 subdomains, S1, S2, and S2 prime, and these domains are responsible for the binding, interaction, and entry of the virus particle. And most of the vaccine are designed against the epitope of this virus. So for the especially immunologists, this protein is very important because of the vaccine development. Another important protein is the RNA-dependent RNA polymerase. This is one of the major enzymes that essential for the viral life cycle, and this is encoded by the NSP12 gene. And you know that this is the responsible for the genome synthesis of the new viral particle, and that's what the most of the drugs targeted against. They target this enzyme. And the remdesivir, you might be knowing, that is actually inhibiting the RDRP activity, and that's what it is possibly inhibiting the viral infection. Again, it is also very similar to the other coronaviruses. That's what the remdesivir are showing, at least you can say the admirable benefits to the coronavirus patient because of their nature to inhibit the activity of RDRP. The third very important enzyme is the main protease. This main protease have tendency to leave the viral particle synthesize longer polypeptide chain. It leaves into the fragment, and that help into the viral assembly. So the basic idea is that if we'll be able to interfere with the activity of this main protease, we could be able to control the viral multiplication. There are two more advanced reasons for selecting this main protease. One is that it is showing 96% similarity to the other viruses, and second is that the human gene is devoid of this kind of gene, so it is entirely different from human gene. So that's what, if you synthesize any compound, if you target by any compound to this protein, it won't affect the host, and that's what it may not be toxic to the human, and that's what the drug molecule developed against this enzyme could be the very, very beneficial. So taking consideration into this structural information of the main protease, we start our work to search the best molecule or potential inhibitor of this main protease. Now the question that I want to summarize that the drug development procedure is very tedious. It is time consuming. It takes a longer time, and even after getting a successful molecule, it has to go through the clinical trial, and through the clinical trial, it has to cross a barrier, and then it comes to the market. So the disease, pandemic disease like coronavirus, you need emergent option. You need emergent medicine. So in that case, drug repurposing is one of the technology through which we can use the already known drugs that is synthesized for the made for the any other diseases. We can try that drugs to stop the multiplication of coronavirus. In that context, the drug repurposing, since the already FDA approved drugs, so there is no cytotoxic issue, and we have to shortcut many steps, and that's what, if you are getting the admirable inhibitory potential, it could be implied for the control of pathogenesis of SARS coronavirus. In this context, we try to start the drug repurposing using the structural feature of the SARS coronavirus. And recently we published this paper, and this is a cover image of the Bioscience report where they took our image, because this compound are showing very excellent inhibitory potential to the main protease. So for that, what we did, we used the following approach, like we analyze the crystal structure of a main protease. then identified the critical residue responsible for the activity and what are the residue involved in the binding to the known inhibitor. Then we perform the high throughput screening against the FDA approved drug database because we screen only among the drugs, followed by the molecular docking and then the structure analysis for the final selection of the best inhibitor. While doing so, we selected out of 2000 compound we screened, we selected 10 best compound, and these compounds are showing excellent binding affinity, and the binding affinity actually determined in terms of the binding energy, that is called the binding free energy, and as the negative signature and with the higher magnitude showing the excellent binding activity, and generally -7 and above is considered as a good binding affinity. And we found that these compounds are showing very good binding affinity. Not only the inhibitory potential we have also determined, and then we determine the inhibitory potential of all these compounds. After that, we perform the ADMET analysis, I mean the drug likeness analysis further to select the best compound, and all the drug likeness analysis we have selected galaprevir and marviroc as a potential inhibitor of this molecule. This is the structure showing. I can answer you later because so you can see the three-dimensional structure where it is binding. You can show the ligand are binding deeper inside the cavity of this main protease, and they are stabilized by the many interactions, especially the cysteine 145 is playing important role in the activity, and you can show this compound are binding to the cysteine 145. So binding to the active site residue is generally proposed that if an inhibitor binding to the active site residue, it potentially inhibits the enzyme activity, right? So the second compound, you can see the binding pattern of the second compound marviroc. It is also, you can see there are some bigger insides and it penetrated deeper inside the cavity, and we propose that this kind of inhibition, this kind of binding could potentially inhibit the enzyme activity. And then based on that, this paper publication published, and now the Brown University in USA, they took this compound for further doing clinical trial on the patients, and the clinical trial on the patient have already been started. Let's hope for the getting good results so it can comes to the market for the treatment of the coronavirus diseases. Now the second aspect of this disease, because side by side we started to search the new compound also, so we use the pharmacophore based screening, means that we first developed the pharmacophore modeling and then followed by the virtual screening. So in this context, what we did, we analyzed the crystal structure of main protease published with the ligand, and then after analysis of the ligand we generated a pharmacophore, and through that pharmacophore we searched in the PubChem database the compound showing more than 9% structure similarity, and then these compounds were selected and for the screening, and through that out of 409 compound we performed the ligand based virtual screening. We selected 172 compounds, then we performed the docking based screening, then we screened to the 53 compounds. After that we performed the ADMET analysis. ADMET analysis is showing the drug likeness characteristic. So then we selected 5 compounds, and finally with the 5 compound we proceeded for the MD simulation to check the binding affinity and all these parameters. So you can see here, this is the crystal structure of main protease published in Science, and this is the inhibitor. It is in the crystal structure. You can see here in the active site cavity, it is known as N3-ILP, and this is the interaction pattern of the N3-ILP inhibitor. So we generated the pharmacophore of the N3-ILP. You can see here the binding pattern, 2D map of the binding pattern of N3-ILP, and we generated the pharmacophore model, and based on that pharmacophore model, we searched the compounds sharing 90% pharmacophore features similar in the PubChem database, and through that we have selected the top 20 best compound, and you can see here their binding affinity are quite admirable. And not only this, you can see the structure of this compound, these 5 compound, and this is the N3-LP inhibitor. So all these 6 system was taken in consideration for all the binding interaction and all the analysis. You can see here these are the ADMET properties, because for a drug-like molecule you have to verify all the parameters in drug likeness, and we checked all the parameters are verified almost good for all the 5 compounds. Now the docking analysis give you the static picture that how the compound bind, but there is possibility of the many pores of the binding, many ways of the binding. That's what the docking analysis always complemented with the molecular dynamic simulation, and that give you the dynamic picture. So the through molecular dynamic simulation, you can see we check the root mean square deviation, radius of gyration, and accessible surface area. All these analysis showing that these compounds are behaving as the best inhibitors are behaving. Like, I'm not going to explain you in the detail of the theory behind it, because I prefer to show you the work, and if you have any question later I could be able to answer. Then we also perform the RMSF analysis. RMSF analysis is the root mean square fluctuation, that what are the residue, what are the region of this protein is actually responsible for the alteration in the activity, binding, and accommodation of the ligands. And you can see here, these are some residues showing higher fluctuation while binding to the ligands. Then we perform molecular mechanics based calculation of the binding energy, and you can see there are different types of energy like the delta E van der Waals, electrostatic, gaseous, delta G polar, non-polar, solvation. These energy are estimated for all the compounds, and through the energy dynamics it is clearly showing that these compounds are behaving like excellent inhibitors of main proteases. Then another very important factor is the hydrogen bond. In the previous slide I shown you that the hydrogen bonds are formed between these two protein one with the ligand and the hydrogen, but from the docking you get the static picture again whether this hydrogen bond is maintained throughout the simulation trajectory or not. So you can see that all the hydrogen bond are maintained throughout the trajectory, means that this hydrogen bond is stable and forming a stable interaction, forming a stable complex, and that's what the probability of inhibition of the enzyme is very high as the hydrogen bond maintained. And after the simulation, we again took the picture of all the compounds with the protein, and you can see the binding pattern, binding pattern, binding pattern of all the enzymes, and you can see here that they maintained before and after the same number of hydrogen bonds are maintained. Then one of the more parameter we generally analyze the secondary structure analysis, that whether the binding of this compound alters the secondary structure or not, and we can find that all the compounds are doing the minor fluctuation in the secondary structure as they expected for the potential inhibitors. So this is the conclusion that the two compounds, especially the compound, this is the ID given that exhibit the best activity and delta G is excellent, all this way we proceeded. Now we further extended this study to take the more crystal structure to get the more confined inhibitor, more high quality inhibitors, because in the previous case we have taken one crystal structure and then based on that we generated a pharmacophore. Now we took the 121 crystal structure published in the PDB and take the best 10 compounds showing the best inhibition to the main protease, took that compound, generated the pharmacophore, and then we search the PubChem database, about 1 billion of the database compound having 1 billion PubChem database having 1 billion compound, where we selected the compound showing 90% similarity, structure similarity, and then based on that 10,433 compounds were selected, and then we performed admit analysis, then again filtered the library, then performed the finally selected we selected 5 compounds for docking analysis, and among them 2 best compounds were selected for the anti-inhibitory mechanism analysis as well as the MD simulation. This is the list of 10 PDB ID that are showing the excellent inhibitory potential or excellent binding to the main proteases. From that, we perform the high throughput screening. Finally, the 5 compound top compound that qualifies all the parameter, both binding parameter as well as drug likeness parameter, were selected for the analysis. This is the docking result showing that how these compounds are bind. You can see here all these compounds bind to the active site cavity, deeper inside the active site cavity, and then we performed structure analysis individually, and you can see the interaction pattern of each the inhibitor with the protein's active site, and these all are showing excellent number of hydrogen bond interaction, van der Waals interaction, and other hydrophobic interactions. We check whether these are stable or not, and you can see that especially we took the two compounds, best compounds, and you can see here these two compounds showing excellent RMS, DRG, SASA value, and also the expected density where it is showing binding. There are the maximum fluctuation are noted that it has showing as penetrated towards the activity. Then again, we checked the hydrogen bond analysis and we found that about 220 hydrogen bonds are maintained throughout the trajectory. Then we added some more analysis like the principal component analysis that gives you again the stability of protein ligand complex, and through the principal component analysis, we also found that this complex is very much significantly stable, and also gives free energy landscape showing the better energy profile. And based on all this analysis, we conclude that these two compounds, topimide is 1042563 and is another is 7114, showing the highest binding affinity, validated all the drug likeness parameter, and proposed as an excellent inhibitor for the SARS coronavirus main protease. And this paper recently published in the virus research. So at last, I would like to thank to my students, my team, Dr. Taj Muhammad, Dr. Shama Khan, Dr. Saleh Anwar, and Anas Shamsi. These are the four guys involved in all the works. And at last, I'm thankful to all the audience and especially Dr. Rashmita Samal for inviting me. Now you can ask if you have any question. I'm happy to answer.
For this insightful presentation, Dr. Imtiaz. You have nicely shown us how drug repurposing could be one of the best choice for therapeutic management. We have a question from our Pro Vice Chancellor.
Yes, sir.
Thank you, Dr. Imtiaz, for an excellent presentation and very optimistic results on the fight which we are doing with Corona. I had a question because there are a lot of reports of some natural products prescribed by Ayush as well as many other competent companies proposing for this particular pandemic. So whether there is any literature or any work which you would like or anybody is doing regarding its binding pattern or its way by which it can really inhibit the mechanism which you are explaining.
Yeah, there are two. Initial slides I told you that many ways to target the coronavirus infection. The major two aspect is either we do the structure-based drug discovery, like we target the inhibition of the particular enzymes. Another very important target is the cytokine storm, where our immune cells get compromised with the situation and the higher, the fever and all those things, the alteration in the cytokine profile, all these things. So as of current literature, most of the natural products are showing good impact in that way because it acts as an immune booster, like the vitamin C, like you can see the Gilo is again having a very good vitamin C content and all the and immune booster. So as an immune booster, they are showing excellent inhibitory potential, excellent activity against the coronavirus. Now come to the structure side. Chinese herbal medicine, one of the well known in the China, and it is one of the very big database of the natural products. India also have a database, but not as too big. It is by the CMAP. So people are trying to explore the mechanism. Many papers are published that showing that a natural product inhibits the activity, but so far none of them are verified. I mean, verified to the next level. At the level of computational method, it's fine. Many people shown that these compounds are showing good activity. Many reports. You can, if you Google it, you can or you PubMed, at least about 1,000 paper published on these different aspect of the natural products. But again, everybody is, because the time is the main constraint, so everybody trying to extract the mechanism on what way it is inhibiting, whether it is working at level or not, like that, or at the in vivo level. But of course, it has a very good hope because the many natural products have the potential.
Okay, thank you, Dr.
Yeah.
Thank you,
Thank you, Dr. Ilkia. So one of
My
our
pleasure.
participants, so he wanted to know, or your view, particularly he wants your view on this.
Yeah.
So he wanted to ask, in India, though some of the population are infected by COVID-19, but they got cured without any prolonged treatment. But in case of other Western countries like UK or in Australia, so they have second wave of infection. So what could be the possible reason?
Secondary infection you're talking about, or you are comparing the Indian population with the American or the Western population?
Yeah,
There are two questions,
yeah.
perhaps.
So yeah, he has an observation based on that
So,
he asked. Means
yeah,
in
yeah, I am answering both the question. One question is that different severity of disease in the different person, or irrespective of the region and all these things. The reason is that it all depends on the cytokine profile of your body, because how our body react to the infection. And I told you initially that the cytokine profile is high variation in the person to person, and that's what you can see even the case of the chloroquine and hydroxychloroquine is somewhere it is failed and somewhere it works well. The reason is that the difference in the cytokine profiling all patient to patient. Second is the secondary infection you are talking. It is actually the people are many reports are coming that this virus are mutating fastly, and the new sequence genome sequence showed that the some variation occurred in the virus. So maybe the chances of mutation causes the new strain, development of new strain that may cause the infection. But in most of the cases, if the one person get infected once, then the chances of the recurrence of infection is very low.
okay. So one thing I would like to know, like receptors plays
Yeah,
very important role in recognizing this
yeah.
virus. Virus also hijack them. So do you feel there is some variation? Maybe there could be some variation among the what is the Asian people compared to the Western countries people who are more susceptible.
Yeah, possibly because even that may be possible, and also the affinity, because the affinity of the spike protein to the receptor varies depending on the sequence of receptor as well. But I think not much studied so far, because it should be studied in the prospect of the functional genomics or structural genomics, that how much sequence variation. But of course, yes, it has some role in the chance of the, not even not in that. If you see the smoker and non-smoker, the
Okay.
level of expression of S2 is different. The smoker are high prone to get infected with the coronavirus, because the the expression of S2 high in the smoker. So there are many ways to understand the situation.
Okay. One of our participants, he would like to know your opinion on plasma therapy.
Yes, plasma therapy is one of the oldest method, traditional method, and you can't say it is the, well, it is just like some support if you are having nothing in hand, because you are giving the antibody outside the system. First, second thing that you are not generating the new antibody. Actually, if you see the whole immunological profile, the amount of antibody generated in the person is depending is by their own B cell and T cell accordingly. So the phenomena goes on. So based on the demand, our body produce antibody against the infection. But in that case, you are giving from the outside. So of course, it will initially neutralize the viral load, and somehow it helps, but it is not a permanent cure. So again, it is has a large variation. Somewhere it works well, somewhere it won't work well. So it is not, you can say, the permanent cure. But yes, it is at least whatever the medicine available in the market for the coronavirus, all are the having the certain advantage. That's what people are trying. Like even stem cell therapy is showing very good results, because it is more and more efficient than the any other thing. But because applying a stem cell therapy is a big task. So yes, plasma therapy is having role, but not it is the replacement of anything. You can say.
Hello.
A complete solution.
Yeah, yeah,
Yes,
right.
sir. Hello.
Yes, tell me.
Yes, sir, this is my question. So I was just want to ask because ICMR denied this because it could cause of death. So how it could cause for death because it is just like similar to the blood transfusion. So how it could cause for death to a patient? Plasma therapy.
No, they actually, it is a very complex issue. Okay. So
Yeah.
I told you that actually the plasma therapy is given to those patients which are at the, I mean the mm, stage of about to death or you very, I mean the highest stage of the infection, right? So if the patient's immunity and the system is itself weak, and in that case you are giving the extra, at least whatever the plasma you are giving, it is from the other's body. So there are chances of cross reaction are also there, and if it fails at somewhere, then automatically it may lead to the death. That's what the people are trying to avoid. But since you don't have any option, in that case it gives you certain help. You take it either positive way rather than negative way.
Okay, sir. Thank you, sir.
So
I got
one,
my
we
answer.
will take one more question. So
Yeah,
the one participant he want to have a opinion means about vaccination in India means what is your opinion whether we will be getting those vaccine in next few months or is it possible or not?
no, ideally it is not possible because the for the vaccine development it puts about 13 months at least to do all the formalities. I don't know, you have, might heard the news that initially
Yes,
Bharat
Bharat,
Biotech announced
Bharat
that on the
vaccine.
15th of August, yeah, we are going to release, but again ICMR denied it is not possible. The reason is that since this disease is dangerous, and that's what many cares. I mean, for all the disease vaccine development, you have to take a proper care, and proper validation is required. So until unless the proper validation required, it can't be possible to come in the market. Yes, the two vaccine is about to come in the market. One is from the Oxford University that are the best vaccine as of the review right now, and one from the Russia. But still, none of it in the market. So actually, the validation time is always needed because you have to test. I think yesterday news that the ICMR allowed to test the vaccine. I mean, start the vaccine trial in AIMS. Now it is allowed to test, but after testing it have a lot of timing to check on the volunteer all these things, then it will come market. But yes, we should have good hopes that like the other vaccine it could come earlier. That's the possible.
So thank you very much, Dr. Imtiaz. And so your work
Thank you
is
very much.
published.
And
So this work is published
I mean,
already.
yes, the first one is published in the BBA, one of the leading journals. Second is published in the Bioscience Research, Bioscience Report, and the third is published in the Virus Research.
Okay, then the
And few
participant
more
can.
I'm waiting. Yeah.
Okay, okay, sir. Thank you very much,
I
sir.
can share the if you need. I can share the paper for the audience. They can.
Yes, sir. That would be a great help.
Yes. So I can share to this link itself, so the student can directly from there.
Thank you so much, Dr.
Okay.
Imtiaz, for such a
Yeah.
wonderful talk and having a great overview on this particular topic. And congratulations on the amount of work that you have put in on coronavirus. Even though it is just 5 or 6 months
Thank you.
old, you have published so many papers and shared us so much of information today. I'm sure that that this session has given a lot of information to all our participants. Um, Thank you very much and stay safe. Dear audience, now this was the last talk of our session. I would just take 5 more minutes. I know that you people are hungry now. 5 more minutes to wrap up the session. So we would request Dr. Durai Pandey, Coordinator Department of Life Sciences, to deliver the vote of thanks.
Am I audible?
Yes, sir.
Thank you, Dr. Asmita, for giving this opportunity. Good afternoon, everybody. First of all, I would like to thank our Honorable Vice Chancellor, Professor H.M. Maheshwaraya, for giving permission to organize such a webinar on COVID-19 and for his grace on this occasion with his inaugural address. He is truly inspiring to all of us. And I extend my heartfelt thanks to Professor G.R. Nayak, Pro Vice Chancellor, as well as Dean School of Life Sciences. So he is the inspiration to all of us, and thank him for his constant encouragement in organizing such events. And in fact, he was literally sitting right from the preparation of invitation and sitting till now, listening the talk. Thank you, sir, for all your help that you rendered towards the Department of Life Science. And I must thank our Registrar, Professor Mustaq Ahmad Patel, for his logistic support as well as his presence for this meeting. Thank you, sir. And I extend my thanks to invited speaker, Professor Barbara Broker from Germany, for her inspiring and in fact inimitable and lucid talk. Remember, she is attending from Germany at morning 6 o'clock in her country. Thank you, madam, for your dedication and joining with us today and sharing your research with our students as well as the participants from rest of the country. Thank you. And also I extend my heartfelt thanks to the other speakers, Dr. Shashidhar Manda, as well as Dr. Imtiaz Hassan, for their wonderful talk and in fact their wonderful research that they have done in this past 6 months. Though you know that everybody is sitting home, okay, enjoying with TV, but there are researchers who sat in the lab and doing the work for us and also sharing their work with us is truly inspiring. Thank you, sirs, for your informative talk as well as giving the current statistics about COVID-19. Thank you. And also I must thank in fact Dr. Rashmita, because she is very proactive in organizing such events. And in fact, she has purchased this Google Zoom from the US and did a lot of homework for organizing this event. And it's truly astonishing. Thank you, Dr. Rashmita, for organizing such event, and of course, many more to come in the future from the Department of Life Science. I extend my thanks to Dr. Kavi Shankar and Dr. Rakesh Kumar Sharma for their wonderful support that they are delivering to the Department of Life Science since from their joining. Thank you. And last but not least, I must thank all the participants from Central University of Karnataka as well as from rest of India, because this type of event is organized for you. So your presence is more important. Thank you so much for joining with us and listening to all the talks. And I forgot, I have to thank Mr. Vinod as well as Mr. Manjunath for their logistic and technical support for organizing this event. I'm pretty much sure that all of you would have enjoyed with the coffee as well and the informative talks from all three eminent speakers. Thank you one and all. Have a safe day ahead. Thank you.